In this video, we're going to be studying the reproductive system in the lab manual, which starts on page 221, although the material really starts on page 222. The first thing they start to discuss is just a really basic. There are several different kinds of reproduction. There's asexual reproduction, which has several subtypes, and then there's sexual reproduction. There's not a human on the planet that can do asexual reproduction, but there are lots of other animals that can. Including vertebrates. There are some lizards that can do asexual reproduction. A lot of other more primitive animals like sponges, they rely on asexual reproduction. And so it's not something that is unheard of in the animal kingdom. We're just not going to spend a lot of time talking about it. Since humans do sexual reproduction, that's where we're going to be spending most of our time. And sexual reproduction is defined by the formation of gametes, which are eggs and sperm, and then the fusion of those gametes to create a zygote that will then develop to make an organism. We're going to be starting with the male reproductive system. It's designed to make the sperm, which is the male gamete. The process that makes the male gamete is called spermatogenesis, and it involves both mitosis and meiosis, but mostly meiosis. There's a picture at the bottom of page 223, and I'll show you a similar picture here in just a second. But the process of spermatogenesis occurs within the testes, which are the male gonads, within the testes we have these small tubes that are called seminiferous tubules, and within those tubules we carry out this process to actually make the sperm. Around the outside of these tubules we have stem cells that are called spermatogonia. Those cells have 46 chromosomes, just like most of the other cells that we have in our body. They divide by mitosis, and when they do that, they're going to create two cells. They're both exactly identical to each other, but one of them is going to stay behind and still be the spermatogonium. The other one is going to differentiate and start the process of meiosis, and that one's going to be called a primary spermatocyte. That primary spermatocyte still has 46 chromosomes inside of it. When that cell starts the process of meiosis, which P.S. if you don't remember meiosis, it's something that we discussed in the first half of biology, and so go back and kind of review your notes, but it has two rounds of division, meiosis 1 and meiosis 2. During meiosis 1, we separate homologous chromosomes, and during meiosis 2, we separate sister chromatids. We end up producing 4 cells when we do meiosis, and each of those cells is genetically distinct from each other, and they have half the number of chromosomes that the original parental cell had. So by the time our primary spermatocyte undergoes the first round of divisions, we will have made 2 secondary spermatocytes. Each of those secondary spermatocytes does the second round of meiosis to each make 2 spermatid. So we're going to make a total of 4 spermatid. The spermatid are immature sperm, but they have half the number of chromosomes, so they have 23 chromosomes inside their nucleus. And they're eventually going to mature into actual sperm cells, which are also called spermatozoa. So here's what that kind of looks like in picture form. The spermatogonium is around the outside edge of the tubule, and it divides by mitosis to make another spermatogonium, and then what's going to become a primary spermatocyte. It divides to make 2 secondary spermatocytes. Those divide to make 4 spermatid, and then those are going to mature and grow their nice little flagellum so that they mature into spermatozoa. The spermatid and the spermatozoa are both haploid cells, which means they have 1 set of chromosomes, which for a human, again, that's 23 individual chromosomes. So you guys have a similar picture to this. It's figure 17.1. All right, flipping the page. So if we were to do a biopsy through a testis, this is what we would see. This is a seminiferous tubule. There's part of one over there, part of one over there. Since these tubules are circular, we've got space in between. That space is filled with cells called interstitial or Leydig cells. Your lab manual calls them Leydig cells. Now the testes are really designed to do 2 things. Make the gametes, that happens in the seminiferous tubules, and make testosterone, and that happens in the interstitial or Leydig cells outside here. It says to look at a slide of the seminiferous tubules and to draw and label the structures of the seminiferous tubule in the space. So we're going to do that using this slide that I have pictured right here. So let me get my label and pen on. Okay, so first off, do yourself a little circular doodle and then label that whole doodle the seminiferous tubule. Outside, we'll go ahead and draw a few cells. Those are the Leydig cells, which most textbooks call interstitial cells. I don't know why I made that an X, Leydig cells. And then within the seminiferous tubule, we have spermatogonium around the outside edge. So good luck spelling that. Spermatogonium, especially given that my handwriting is miserable on a slide. Those are going to start that process of division. So the next cells in are going to be primary spermatocytes. I'm going to run off the edge of my slide soon. Okay. They divide to make 2 cells. Those are secondary spermatocytes. I'm going to start abbreviating. Each of those divides to make cells. Those are spermatid. I won't abbreviate that one. And then those mature, and you can kind of see that better off over here, but they mature into spermatozoa, or just sperm, and those are the cells that have the flagella, which are all those things going on there. And so these tubes are what are actually generating the sperm cells within the testes. From there, figure 17.2 gives you the basic anatomy of a sperm. There's a couple of different parts that really matter here. One, the flagellum, that's what's going to allow them to swim so that they can find the egg and then fertilize the egg. The mid piece has a bunch of mitochondria in it. If you remember from the first half of this course, mitochondria generate energy. A flagellum actually requires a lot of energy in order to power it, and so that mid piece is going to be supplying that for us. Within the head, and I did not draw this in the picture, so I'll doodle it right here, there's a nucleus. The nucleus is where we're going to have those 23 chromosomes, so that's the nucleus. I'm just going to abbreviate. And in front of that, there's this little bag of enzymes called the acrosome. Those enzymes are going to be necessary for breaking down the barriers that surround the egg once the egg has been ovulated. And it's going to be critically important that we have lots of sperm to release lots of those enzymes from the acrosome so that we can do that. Now, on the top of page 225 in your lab manual, it says, look at a slide of a sperm on a microscope. Instead of doing that, I'm just going to have you guys doodle the picture that I have right here. If you need to pause the video to do that, have at it. All right. From there, we start to get to the macroscopic or, easily observable features of the male reproductive system. So to run you through what we've got going on here, we're going to start in the gonad. The gonad is the testes. There's 2 of those in most men, a right and a left one. That's where we have those seminiferous tubules to make the sperm and the Leydig cells that are going to make the testosterone. Once the sperm are created, they are then going to move to this little comma-shaped tube that's called the epididymis. That's what I like to call sperm summer camp. That's because in summer camp when I was a kid, I learned how to swim, and that's really the one thing that sperm have to be able to do is learn to swim and swim to the egg, and they learn to do that or mature within the epididymis, and so sperm summer camp. If a man does not ejaculate, sperm will be recycled within the epididymis. If a man does ejaculate, from there, the sperm is going to enter this next tube, which takes like the most circuitous route to get where it's going. It starts down here. It goes, go away little box. It goes around the front of the pubic bone, over the top of the bladder, around the back of the bladder, and then it merges with another little tube. And so all of this is all the vas deferens, sometimes called the ductus deferens. The vas deferens is just going to carry out some peristaltic contractions to push the sperm closer to the exit point. Your fun little fact, vasectomies cut and tie those 2 tubes, and they do that right there. Because it's really easy to get to the vas deferens within the scrotum, and so they'll cut and tie those tubes. That means sperm are still made, testosterone is still made, but because those tubes are tied, the sperm have no way to leave the body, and that's why a vasectomy can end up creating sterility in a man. From there, I'm going to change colors really fast, in theory really fast. This little structure right here that's all lumpy and bumpy, there's another one on the other side. Everything that I've mentioned so far, there's 2 of those, a right and a left. So there's another seminal vesicle on the other side as well. The seminal vesicle is going to be contributing fluid that has nutrients in it so that the sperm are going to be able to survive longer in the female. The seminal vesicle and the vas deferens merge together to create a very short tube, which there's still 2 of, and that tube is called the ejaculatory duct. That duct merges with the urethra, and that's where we go down to having only one of something. There's only one prostate gland that goes around the urethra, and then there's only one urethra that's going to allow the sperm to leave the body. Now, in addition to that, there's also, which your lab manual calls that a Cowper's gland, but there's also a bulbourethral gland. the seminal vesicle, the prostate gland, and the bulbourethral gland all contribute fluid that's going to add to the volume of semen. So if you think about what semen actually is, it's sperm plus the fluid from the seminal vesicle, the prostate gland, and the bulbourethral gland. So that's what semen actually is. All right. Other stuff that I haven't talked about just yet. So the urethra, as we're leaving, it goes through all of this erectile tissue, which is blue in the picture, and it's just labeled erectile tissue in this picture. But we have what are known as cavernosal bodies. There's two of those, and I'll show you a different picture that kind of labels that later on. But this is the cavernosal body, or Corpus cavernosum. And then around the urethra, all through here, this is the spongy body, or Corpus Spongiosum. That tissue, when a man becomes aroused, it fills with blood, and the blood pressure gets so high that it makes the penis erect. And so that's why it's called erectile tissue. Seen in some men, but not all, the prepuce is the foreskin. So if a man has been circumcised, that has been removed. All right, let's see. Top of page 227, the lab manual talks about benign prostatic hyperplasia. It is one of the common things that develop in men who have reached a certain age, we'll say. What happens as men age is the prostate gland enlarges. It's not cancerous, that's why it's benign. However, it is uncomfortable and it makes urination more difficult. And that's because what it's doing is it's constricting the urethra so that it makes that urination more difficult. It can also make sperm exit more difficult for that matter too, but men are going to be less troubled by that than they are the frequent urge to pee and the difficulty with actually peeing. There are treatments for this. It kind of depends on where a guy is and how problematic it's being, but there are various drugs that we can use to try to reduce the size, and if worse comes to worse, there is some surgery for that. But it can cause urinary retention, and urinary retention can be life-threatening if it's not treated. So even though it says benign, it's still something that needs medical treatment. All right, underneath that picture, it talks about the erectile tissue in the penis just a little bit more. So this is, if you were to do a frontal section through the penis, that means you sort of cut it so that there's a front, pardon me, and a back half. I've always kind of thought it looks like Cartman from South Park when you look at a cross-section of a penis like this. But the two eyes, those are the cavernosal bodies, or corpora cavernosa is the Latin term for that. And then the mouth itself is the urethra, but around the mouth you have the corpus spongiosum, or the spongy bodies. This is again the erectile tissue that's going to fill up with blood. So I'm going to get sort of weird for just a second. I think I'm going to show you a video for this and lecture once I actually get to it. But we've got these arteries that are in the eyes, and then there's more arteries that are up here. Those arteries bring blood into the penis when a man starts to get aroused. And then, essentially, we vasoconstrict the vein so that the blood can't leave the penis when a man is aroused. And again, what that's going to do is it's going to create hypertension within the penis, or very high blood pressure. And that's going to make the penis stand erect. So an erection for a man is a blood pressure issue. There's not a bone and a boner, in other words, is kind of what I'm saying. So my weirdness that I'm going to get to is there are lots of drugs that can be used to treat what's known as erectile dysfunction or impotence. Those drugs were originally actually designed to treat people by lowering their blood pressure. And it turns out those drugs make a penis erect. And so they found out there was a much bigger market for it in terms of erectile dysfunction. That's what Viagra is. It was a drug to lower blood pressure that had the side effect of making penises erect. And then it turned out that side effect was actually worth more money to them than the lower blood pressure and medication. So there you go. That's what Viagra and Cialis and whatnot do. Now, here's the other thing. If you've ever watched any of the commercials or heard any of the commercials for any of those drugs, they say if you've had an erection that lasts longer than 4 hours, you need to seek emergency medical treatment. If you have an erection that lasts longer than 4 hours and it's unwanted, that's called priapism. Priapism means that this vessel that we have closed doesn't open back up like it's supposed to. And as a result, the blood stays pooled in the cavernosal bodies and the spongy body. And if blood stays in one place for too long, it clots. That clotting can prevent the penis from emptying once that blood vessel does actually start to open back up. And so that's why this is a medical emergency. If the erection lasts too long, your blood clots, and then that can lead to lots of problems, including death of the penis. And so please don't use Viagra or Cialis unless you have been diagnosed with a condition that requires that as a treatment. All right, next. I'm going to switch on over to the women. So while men carry out spermatogenesis, women carry out oogenesis. And we have a very similar process that happens, but there are some differences. Where a spermatogonium can eventually make 4 functional sperm, an oogonium will not make 4 functional eggs. It will only make 1 functional egg, and then it will make 3 garbage shells called polar bodies. So here's the process. The oogonium still divides by mitosis, and when it does that, it makes another oogonium and something called a primary oocyte, which is analogous to the primary spermatocyte. The primary oocyte starts the process of meiosis before a woman is even born, and then it freezes. And then when she hits puberty, 12 years later or however long it is for her to hit puberty, then that primary oocyte will unfreeze and start the process of meiosis back up. And so what that means is, unlike in men, there are some problems that can arise in female egg development because our cells have been frozen in a state where the chromosomes are glued together in synapses. That means women are more likely to suffer from non-disjunction, which is failure of the chromosomes to separate during meiosis, especially as she gets older. A woman who is 35 or older who gets pregnant is considered to be what's known as a geriatric pregnancy because she's at a higher risk of having complications for herself and a higher risk of chromosomal abnormalities in her baby because we froze our eggs during the process of development inside of us and then didn't continue that process until much later on. So once we hit puberty, one of the eggs and one of the follicles and one of the ovaries every month is chosen to go ahead and continue the process of development. The next month, the other ovary will choose a different egg and start that process in that one. So our ovaries take turns. That's very different from how sperm do it and testes do it. Testes just make bazillions of sperms and release the bazillions of sperms, and they don't start until puberty, and then they continue till a man dies. We are much more complicated in terms of how we're going to be making those eggs. So if we're going to be releasing an egg, it's going to continue that process of meiosis, and the primary oocyte is going to divide in the first round of meiotic divisions to create a secondary oocyte and then a polar body. A polar body is a junk cell that donates its cytoplasm to the egg so that the egg is going to be bigger. Now it's the size of 2 cells instead of just 1 cell. The secondary oocyte is what actually gets ovulated from an ovary, which means we release an egg before we're even done making it from the ovary. If the woman does not have sex, the secondary oocyte is just lost with the menstrual flow and it never finishes development. If, on the other hand, a woman does have sex and a sperm fuses with a receptor on the egg membrane, in other words, fertilization occurs, then the secondary oocyte will finish meiosis and create even more polar bodies that are going to sacrifice their cytoplasm to make the egg even bigger, and then an actual functional egg. So women can only make functional eggs if we've had sex and a sperm have entered the egg. Otherwise, we just get rid of an egg that was partially finished but not 100% finished yet. So here's what that picture looks like in us. This is figure 17.5 on page 228. So notice, before birth, we've got our oogoniums in place. They started meiosis to, let's see, right down here, to create our primary oocytes that are rested in a very early stage of meiosis. We're then born, we go through our first 10 to 12 years of life, and then when we start puberty, we continue again, and the primary oocyte goes ahead and it finishes that first round of meiosis to create the secondary oocyte. And then the secondary oocyte will end up making some more polar bodies and a functional egg, but only if the sperm actually gets inside of there. So yeah, more complicated, lots of breaks, but we end up making a much bigger cell because we sacrificed 3 other cells to make the egg even bigger and stronger and better, essentially. All right, let's see. Top of page 229, you have a simplified picture of an ovary. My picture is more complex. I pulled it from an A&P textbook, so sorry that it looks very busy compared to the picture that's in your lab manual, but mine's more realistic. It adds all the stages. So when we're born, women that is, we have all of our little eggs inside of these little structures that are called follicles. These are primordial follicles right here. When we hit puberty, some of those start the process of development and they become primary follicles. Now, primary follicles get one layer of cells around them. That's how you learn to recognize those. Those cells around the egg, because of hormonal stimulus, start to divide by mitosis, and we get more and more layers of cells around them, so that eventually we've got a developing follicle. This is basically the next thing that's labeled in your lab manual. So you've got primary follicles labeled in your lab manual, and then instead of calling that secondary, the lab manual calls that developing. I need to get my pen because I'm even worse with a mouse. Developing. Okay. Those cells that are around the egg, they end up producing a chemical bath to sort of keep the egg nice and happy, and that creates this fluid-filled pocket. When you can see that pocket, then you know you have a mature, or what your lab manual calls, Graafian follicle. The Graafian follicle, the pressure is going to build up and sort of smush the egg out. That process is called ovulation. And so ovum in the process of ovulation is the next thing that you guys have. Now here's the other issue though. All of these cells that we had around the egg, most of them are still there inside of the ovary. Those are going to end up developing into a structure called the corpus luteum, which is a glandular structure. It's an endocrine gland. That gland is going to produce progesterone in the hopes that sperm find that egg, fertilize that egg, and the woman gets pregnant. If she does get pregnant, the corpus luteum sticks around for about 3 months. If she doesn't get pregnant, it starts to degenerate after a couple of weeks, and then it goes away because she didn't need it because she didn't get pregnant. And so that's what's happening to the ovary as a woman is going through her monthly cycle. There's an ovarian cycle, which is what's happening, what we just described, and then there's also a uterine cycle, which is the menstrual cycle. All right. So just to show you, they didn't really give you slides to look at in the lab manual on this one. They did say look at the ovary model in lab and identify the 5 structures that you had. That was what we just did on that picture. But I wanted to just show you what things look like under the picture. Now, cats are different from humans. Humans release 1 egg at a time. Cats release a bunch because they have litters of kittens, not like humans for the most part. And so cats are going to do more than 1 at a time. But you can always recognize a Graafian follicle because there's going to be that pool of chemicals, and then the egg is going to be sticking off over here where this is the actual egg. These are the cells that protect the egg, and these are the cells that secreted this fluid inside of here. Now this one, it's more immature, but we do still have some of those little fluid pockets that are developing, and so this is just a less mature Graafian follicle. This one is a more mature Graafian follicle. Cross-section through the whole ovary. And they wanted us to make sure we understood the difference between this. So here's a Graafian follicle, and you can tell because there's the egg. Here's a Graafian follicle because there's the egg. But you also have these things that kind of almost look like Graafian follicles, but there's no eggs. Well, that's a corpus luteum, and that's a corpus luteum. This one's a Graafian follicle because there's the egg. There's really not a lot of other things going on here that are late like that. And so the difference between the corpus luteum under the microscope and the Graafian follicle is you can see an egg in a Graafian follicle. You can't see it in the corpus luteum because it's been ovulated out. Now, I've always loved these pictures when I see them. This was taken from a woman who was having a hysterectomy. And she just so happened to be ovulating on the day that she went in for her hysterectomy. So they got this all on camera. So ovary is this white structure that's going on all through here. This is the Graafian follicle that is actually squeezing the egg out. Most textbooks always like to say the egg explodes out. But you can see from the picture, that's not really how it happens. It just kind of oozes out in a sort of gross little, it looks like a little honey droplet sticking off the end of it. But that's the egg that's being ovulated. Now, the cameras used in hysterectomies, they do magnify, but this would still be visible to the naked eye. The egg cell is a gigantic cell because really it's kind of 4 cells in one because of all the polar bodies that sacrifice their cytoplasm. So again, they're just really big cells. All right. So see if you can answer that one based on all the discussions that we've been having. hopefully you can tell that's a Graafian follicle, because there's the egg. All right, here's your macro structures for the female, and I've always kind of hated the pictures because it's like the ovaries are always trying to hide. There's 2 ovaries, just like there's 2 testes, but you can only ever see part of them in the models and in the pictures. So that's the ovary. It's sort of hiding behind the uterus and hiding behind part of the fallopian tube. Fallopian tube is in this picture called an oviduct. In some pictures it's called a uterine tube, but whatever, it's the fallopian tube. It's whatever every doctor I've ever talked to about it calls it a fallopian tube. The ovary, once it releases the egg is going to go into the fallopian tube and then travel to the uterus. Now the uterus has several layers to it, but arguably the most important is this inner lining, which is called the endometrium. The endometrium is the layer that's going to build itself up every month in the hopes that you get pregnant. And then if you don't get pregnant, tear itself down, and it is the reason why we have periods. All that blood comes from the endometrium tearing itself apart. Outside of that, you have the myometrium. This is the muscle that contributes to cramps, if you get cramps, or it contributes to the contractions during labor. Although we do also get lots of other muscles involved, the myometrium is a big part of that. The opening to the uterus is called the cervix. That's the structure that's supposed to dilate to 10 centimeters before a woman starts to actively push during labor. The cervix leads to the vagina. The vagina's got a lot going on, so it's the birth canal. Babies are supposed to get pushed through there. It is very expandable. It's also, so let's say I've seen it described as the site of menstruation, and that's because all the blood and tissue from the endometrium, it comes out through the vagina. So that means like tampons and diva cups or whatever menstrual cup you guys might use, those things get inserted into the vagina to prevent that loss of blood. It is, the cleanest way I've ever heard it described is the receptacle for the penis. That's where the penis goes during sex, or at least that's where it's supposed to. So here's where I'm going to get all weird on you guys again. There is a book out there written by Mary Roach. It's called Bonk. That book describes some of the scientific research methods that have been used to study sex in human beings and just things that doctors that deal with the reproductive system see over time. And one of them is just that the basic education of sex education in the United States and other countries for that matter is so abysmal that periodically there are women in the United States who go in complaining that sex is very painful for them, and it's because the man has been sticking his penis up here. This is not where the penis is supposed to go. The penis is supposed to go here. If it goes up here, that means it's going up the urethra and into the urinary bladder, which 1 means pregnancy is not going to happen if that was the goal of sex, but 2, that tube is not nearly as expandable as the vagina, which means it's going to just be excruciating every time that woman is having sex, and she's going to hate everything that the man is doing. So just make sure you're putting it in the right hole, I suppose, if we're playing that game. All right. Clitoris. We only see a teeny little portion of the clitoris, but it's actually a much bigger structure that is deep to the opening. The clitoris is analogous to the male penis, which means it is erectile. It gets bigger when a woman gets excited. It is a very important structure for the enjoyment of sex. In fact, a lot of women can have an orgasm with just clitoral stimulation, which means there doesn't have to be penetration and an orgasm can happen. So guys, pay attention to this. It's an important little structure. There is also, let's see, I think they label the Cowper's gland for you guys, or the vestibular gland, do they? I'm looking, sorry. I shouldn't be trying to look at 2 pictures at one time. It doesn't look like they did label it in your picture, so never mind, I won't actually label it on yours either, but there is a gland for lubrication. Sex is very uncomfortable for a woman if she is not, quote unquote, wet or lubricated, and so that gland helps with that. Although as you get older, that gland becomes less active, but they make lubricants if you need lubricants. So if sex is uncomfortable for you, please talk to a doctor and make sure you are A, putting everything in the right places, but B, you are making enough lubrication so that you're not actually tearing the vagina during sex, which it's not pleasant. So there you go. Okay. So just questions to ask after the fact. So is this picture showing you spermatogenesis or oogenesis? Well, hopefully you recognize that the picture is of a seminiferous tubule, which means we're in the testes doing spermatogenesis. And so that's what's happening there. I'm always shocked by how many people get this one wrong, because I really could not make it any more obvious. But, you know, penis means male from a biological standpoint. And so this is a male. This is another one I'm always shocked at how many people get this one wrong. The correct answer to this question is E, is what's pointing at the vagina, because this is the vagina right here. Not this one. This is just to show the whole point of the reproductive system is to make babies. And so if fertilization were to have occurred, we would create a zygote, which is happening right up here just a little bit before A. After we make the zygote, about 24 hours, that process that is called cleavage starts, and that cell starts to divide to make 2 cells, and then 2 cells becomes 4 cells, 4 cells becomes 8, and we keep going until we end up with a solid ball of cells right around here. That's called a morula. What happens then is the cells start to migrate until we get a hollow ball of cells, which is happening right around there. That's called a blastula. This process that gets us from here to the blastula takes about a week, and that happens as the egg is traveling down the fallopian tubes towards the uterus, and the blastula right here is what's actually going to implant into the uterus if a woman gets pregnant. So there's kind of debate within the medical community about when does pregnancy start. Does it start up here where an egg gets fertilized by a sperm? Or does it start down here where the blastula implants into the uterus? I am of the opinion that pregnancy doesn't start until we hit this stage. And the reason that I believe that is because there are lots of women that have what is known as, quote unquote, a hostile uterus. And she can make all the zygotes that they want and they can divide like they're supposed to. But because her uterus has an overactive immune system, the blastula never gets to implant, so she will never become pregnant. She will never pop positive on a pregnancy test. She will never carry a baby to term. And so that's why I am of that opinion. That happens more often than you might think. All right. To continue on after implantation, though, we start the process next of what's known as gastrulation. Gastrulation starts to create our first tissues, because here's the thing. All of the cells up to this point, they are all embryonic stem cells. They can all be anything they want to be when they grow up. But once we start gastrulation, they start moving down a specific pathway so that they will become a specific kind of tissue, like muscle tissue or nervous tissue or whatever. It doesn't look anything like a human baby at this point, and yet it is a human baby. We continue that process of making our first tissues. Our first tissues will lead to our first organs. Our first organs will start to make us look more like a living thing. Once we hit around this stage, it's a recognizable embryo, although chicken embryos, mouse embryos, and human embryos all look pretty similar to each other, and so I wouldn't expect you guys to be able to recognize the difference between them. Starting right here is where you start to be able to recognize, yeah, that's a human baby, and so the name changes and it becomes a fetus. Once we hit the fetal stage, at that point, really, all of our organs are in place. They just need to get bigger, and so everything from here until birth is all about the growth of the organs that have already been formed. All right. After that, we've got the actual questions that occur in your lab manual starting on page 232. You have a table that gets you the anatomy and physiology of the male reproductive system, so just pause the video so you can fill in your blanks. Then at the bottom of that page, there's a picture that has some things labeled, and then on the top of the next page, 233, there's a table that relates to that picture. This is what you need for that. Again, pause the video to get what you need. Then questions towards the bottom of page 233. These are all pretty self-explanatory, so once again, pause the video to get what you need. Questions on page 234, guess what? Pause the video, get what you need. Then we'll get to the critical thinking questions where I'll slow down though. Number 1, why are secretions of the seminal vesicles, prostate gland, and Cowper's gland all alkaline? Well, it's because the vagina is a very acidic environment that helps to retard bacterial growth in the vagina to keep it clean. Sperm don't like an acidic environment though, and so essentially men make an antacid to counteract the acidity within the female so that the sperm are going to be more active, and so that's why it's essentially an antacid to help sperm survive better in the female system. 2, if it only takes 1 sperm to fertilize an egg, then why does the average male ejaculate contain about 60 million sperm cells? Well, here's the thing. Yes, we do really only want 1 sperm to attach to a receptor in the egg and fertilize that egg. But here's the thing. When an egg gets ovulated in a human, it's not like the receptors are already available because the receptors are down here. We've got all of these cells that had been contributed from the follicle and the ovary. We need to digest those away. We've got this layer that's sometimes called a jelly coat or a vitelline layer. That has to be digested away. And if you remember, sperm have this little bag of enzymes called the acrosome. Well, it takes hundreds of sperm to release the enzymes from their acrosome to kill the eggs, the cells that are out here, and to digest a path through the vitelline layer so that we have opened up a receptor. Those first cells that do that, by the time they have released their acrosome and killed some of these cells, that kills the sperm too. And so this is not a sperm that's going to fertilize an egg, but it was necessary to help us get through that path. This one failed. We needed it, but it doesn't actually get to fertilize the egg. It's one of the latecomers that actually has gotten here after we've killed off these cells and killed off that vitelline coat that's going to be able to bind to a receptor and then squirt its nucleus into the cytoplasm of the egg and fertilize that egg. So we needed all of those first line soldiers to get us there, and then one could actually go ahead and attach to a receptor and bind. So that's why we need a bunch. All right, number 3. The average female reproductive cycle is 28 days. What happens during this time and how do hormones affect the cycle? A very important note, average female reproductive cycle is 28 days. I know very few women that actually have a 28-day cycle unless they're on the pill, because the pill makes them be on a 28-day cycle. And in fact, most of the women that I know, their cycle is not all that regular. Like it's 32 days one month and then it's 26 days the next month. And so you should never just use, well, it's been 28 days, I should be able to have sex and not have a kid, because that is not a reliable birth control method. That said, going back to the 28-day cycle, during about the first week of the cycle, that's when a woman is having her period. And again, that varies. Some women are lucky enough to only bleed one day. I've always kind of hated those women. Some women bleed for about 2 weeks. And so it varies wildly from one woman to the next. But on average, during the first week, we're having our period. After that, for the next week, we're building the endometrium back up. We do that because of a flush of progesterone that gets secreted. At the end of that second week, that's when ovulation occurs, which means that is when sex can actually get a woman pregnant, is in the middle of that menstrual cycle. The egg gets released or ovulated due to a surge in luteinizing hormones. So if you're ever using an ovulation test kit so that you can try to get pregnant, those test kits are testing for what's known as the LH surge, which is a surge in luteinizing hormone. That happens the day before you ovulate. So when that kit tells you, yes, you're ovulating, it actually means you're ovulating tomorrow because it detected that surge in LH today. After you release the egg is going to start to travel down the fallopian tube, and you're going to continue to produce progesterone in the hopes that you get pregnant. If about a week after ovulation, we do not implant the embryo, then we're going to start to have a decline in progesterone, and we're going to start to complete that cycle all over again. But if we do implant, we keep producing progesterone, and then we stop that cycle. And so this slide sort of writes down everything that I just said. Number 4, how do the different forms of contraception work? Well, there's a bazillion different forms of contraception that's out there, and so it kind of depends on which method you're choosing. So the barriers, that would be like condoms. What they're doing is preventing sperm from reaching the egg. If sperm can't reach the egg, you can't get pregnant. Diaphragms also work the same way. If you're using spermicides, like spermicidal jellies, those are chemical barriers that sort of murder sperm before they can get where they're going. They are often used in conjunction with diaphragms. Hormonal birth control methods. That would be like if you're taking oral contraceptives or the pill. That would be if you're doing shots like Depo-Provera, or if you're doing patches, or if you have implants, like Norplant used to be a brand name for one. I'm trying to think of some others. Some of the others are like IUDs that have hormones. They're called IUCs, like Mirena. All of the hormonal methods, what they do is they trick the woman's body into thinking she's already pregnant. That shuts down her pituitary cycle, which means she's going to stop ovulating. And if you don't ovulate, you can't get pregnant. And so that's how all the hormonal methods work. There are some other methods that are out there, but those are the main two, and so those were the only two that I was going to talk about here in lab. What causes male infertility? A million different things can cause male infertility, and it usually can start with something like his pants are too tight. The scrotum's job is to help the testes hang lower or away from the body to lower the temperature. If the testes are held up next to the body, it makes them too warm, and sperm don't develop correctly if the scrotum does not sag correctly, essentially. So stop wearing really tight pants. That's one thing that can contribute. There are certain drugs where the side effect is you will reduce your sperm quality or quantity. Most notably, that would be chemotherapy. If a gentleman going through cancer treatment had to have chemotherapy, he will very often be infertile at the end of chemotherapy because it killed the testes, essentially. There's a bunch of other things too, but I've just sort of hit the highlights there. Number 6, what causes female infertility? It can be some of the same things. Like, again, chemotherapy is going to cause a woman to be infertile. It can be blocked tubes. Like, if a woman gets an STD like chlamydia, she can very often be asymptomatic and not even realize that she has chlamydia. Well, that means the infection can spread and then become pelvic inflammatory disease. And one of the side effects of pelvic inflammatory disease is scarring in the fallopian tubes, and that can lead to infertility. But I've given you some other reasons that are up there. There's lots of reasons again. Number 7, why does oogenesis only produce one functional egg while spermatogenesis, I don't know why I can't talk all of a sudden, spermatogenesis produces 4? Well, it's because, again, we produce polar bodies when we make eggs, and those polar bodies sacrifice their cytoplasm, and in the process, they're sacrificing themselves. They're going to die. But they do that to make the egg more viable. And so we only make one egg, but it's gigantic. And then the sperm, on the other hand, just do their rounds of division like they're supposed to, and they make 4 functional sperm cells. So that completes the reproductive system. At this point, you should be ready to take your post-lab test.